it in detail today, so don't worry about it. You have not missed anything. But let's just go with. Is everyone joined? No. Two people will still join. Okay. Yes, Arpit. Tell me.
Sir, what are casogamous flowers?
What are?
Casogamous.
Can you spell? What are?
Sir, wait, I'll type it.
Yeah, that will be better. Are you saying casiogamous? It's not. It's called casogamous. Okay. Yeah, it's CHASO. It's called casogamous. Casogamous flowers. So, what are casogamous flowers? Did you figure out what is clistogamy?
Yes, sir.
Yes. And it's not caso. Arpit, I think you're talking about chasmogamous flowers. There is a M.
Sir, my teacher was pronouncing it as casogamous. I don't know what's the correct pronunciation.
Yes, it's not chasmogamy, it's chasmo. So there is clistogamy and chasmogamy. There are two things that I was planning to discuss today. Where's the pen? Yeah. Just one moment. Let me put this off. Yeah. So I told you to go and figure out this one. Can you see this clistogamy? And what you are saying, Arpit, is not chasogamy or casogamy, it's chasmogamy. There's a M also. Are you asking the same thing, or you are like, are we on this? I just want to know if you're talking about the same thing. So there are two things. One is this clistogamy, and another is spelled like this. It's called chasmogamy. So are you talking about this?
Yes, sir, I think it.
Okay. So we'll be talking about both of them, clistogamy and chasmogamy today. Okay, just one more. Anything else you want to ask? We will do this. Did you figure out clistogamy? Yes, Fatima said yes. Was it you, Fatima?
Yes, sir.
Yes. Please tell me what is. Alia is here. Welcome.
Sir.
Good evening. Yes, Fatima.
Clistogamy is like in closed flowers, like flowers which do not open, self-pollination happens. So that is clistogamy.
Yes. Sometimes in some books I've seen they write it as CLEI. Clistogamy. So CLEISTO is also same. So clistogamy you are saying is self-fertilization. So my question is it different from autogamy? According to you, Fatima?
No,
It's not different, it's same.
yes, sir.
Fatima? What do you think?
Sir, I think it is same.
It's same? It's not same. As you yourself said, clistogamy happens in what kind of flowers?
Closed flowers.
Closed flowers. There are some flowers which are permanently closed. They never open. So if a flower never opens, what is the point of making a flower, right? Becomes a question. So if there is a flower that does not open, which means it remains closed. So it is like a big closed system inside which there are anthers and filaments developing. So obviously, can a pollinator reach there if it is a closed flower? Yes, sir.
No, sir.
No. Then what is the motive of a closed flower? How is it helpful?
Sir, protection.
What? Protection.
Yes, sir.
It is actually a very okay. Let's come to these two together. But before that, quickly give me a quick recap of last class, stages of sexual reproduction, and today who will give it? Last time who gave?
Sir, I gave last time.
Okay, let's go to Maharaj. Would you like to give a quick recap of the last class? in front of you on the board?
Okay, sir. Yes, sir. So we started with sexual reproduction in plants, and then sir, first pollination. So basically, sir, pollination is the pollen grains. They transfer from anther. They go to the stigma of the plant. Uh, and then we talked about different pollinators. That is wind, water, and other insects.
Yes. In pollinators, we also discussed abiotic and biotic, right? Non-living components and living components. Correct? Mehraj,
Yes, sir.
I cannot hear Mehraj. Can you hear Mehraj? Fatima, Arpit?
No, sir. Now I can't.
Mehraj, you are not audible. Now you are mute. So unmute yourself. I think Mehraj is having some network issues. Okay, Fatima, would you like to?
So we also did types of pollination. We learned about autogamy, get, geitonogamy, and autogamy is within the same flower the pollination takes place. Geitonogamy is between two flowers but of the same plant. Then we studied about xenogamy. It is cross pollination, different plants, different flowers. Then you asked us to search about cleistogamy, and you asked us to search for examples of each of the pollinations.
Did you figure out the examples?
Yes,
Yes,
sir.
sir.
Yes. So let's start with examples and finish with the examples. Yes. Tell me examples for autogamy.
Sunflowers, orchids, camelina, peas.
Rice, tomato.
Sunflower, orchids.
Yeah,
Yes, sir.
Okay. And
camelina.
pea, right?
Yes, sir. Yes.
Right. And
Camelina.
sorry,
Camelina.
can you spell C?
Commelina.
Okay.
Sir, that's the example of cleistogamy.
Pea, right? And
It
camelina.
shows both of them.
I was coming to that. I'll come to cleistogamy. So pea is a very classic example which shows cleistogamy. Pea and groundnut both. You know, groundnut also have closed flowers. Anyone has seen a flower of a groundnut or a pea? Pea flower you all must have seen. Mendel did a lot of work on pea. Right? Mendelian genetics was done on pea plant. Okay, anyways, geitonogamy. Examples, Fatima.
CUCUR bits, maize and corn.
Cucurbits, maize,
Yeah,
and
Sir,
cotton.
grass.
corn.
Grass is a family. Which grass? Grass is not one. Grassiaceae is a family. It's not like one thing is called grass. Bamboo is also a grass. You know that? Do you know, Arpit, bamboo? The bamboo plant belongs to grass family.
Oh,
Yes, so write it down. Bamboo is a grass, probably the longest grass on earth. Yes. Fatima, complete your examples. Cucurbits, maize.
Corn.
Which one?
Corn.
Corn.
Yeah.
C-O-R.
CORN.
What is the difference between a maize and a corn? Is there a difference between maize and corn? Okay, we'll come to that also. Xenogamy. Tell me xenogamy.
Onion, broccoli, papaya,
Onion, papaya, broccoli.
sir, they are same.
Maize and corn are same.
Yes, sir.
Maize is the plant. Corn is the seed with which you make popcorn. Remember? That is corn. Sweet corn, popcorn. We never say sweet maize, pop maize. But maize is that structure, the plant. Is it clear, Fatima, to you?
Yes, sir.
Yes. Okay.
Excuse me, sir.
Yes, Arpit.
Sir, I just checked this, sir, and it said that, sir, in different regions it is called different with different names.
It is, but basically corn, the word corn is more of market oriented industrial word where you sell corn, but basically you grow maize. When we call about plantations, like from a agricultural point of view, we say maize plantation. So these are maize fields. You can also say corn fields, but corn is that, you know, that structure which we eat, the edible part, that is corn. Corn seed.
Okay.
Okay.
Yes.
Just like we call it like rice, and when we call about fields, we also say paddy fields, right?
Yes, sir.
What is paddy?
Rice.
Yes, but rice, we know rice has grains, the rice like just like corn, and paddy is just like maize in terminology. Okay,
Yes.
the plant is said to be paddy plant and paddy fields. Okay, then we went next. Fatima, you were giving a brief recap. Then the second stage was formation of zygote. Yes.
We learned about formation of zygote or fertilization. Yeah, we learned about that.
What did we learn? Arpit.
Sir, you briefed us about that, sir, pollen pistil interaction.
What is a pollen pistil interaction? Alia?
So,
Alia, can you speak? Are you there? Yes, thank you. What is a pollen pistil interaction, Alia?
When you pollen and pollen.
Is it just me or everyone cannot hear Alia clearly?
No, sir, I cannot hear also.
Alia, you are having some issues with the microphone. Try to fix it if you can, okay? Okay, so we discussed about pollen pistil interaction, and Arpit completed. What is this?
sir, then we discussed about the biological polymer, sir, sporopollen, and sir, later, sir, you explained about the structure of the pollen. Sir, then I left the class.
Okay, so just before you left the class, and after you left the class, we just discussed about the pollen tube. So probably you missed pollen tube. So
Yes, sir, yes.
there are in a mature pollen grain. So let me take over from here, because this is also a part of the question that Arpit asked for me to explain things towards the end. So a mature pollen grain, Arpit and everyone, has two cells, generative cell and a vegetative cell. This I have also told you in the last class. But what I told you in the previous class is that this generative cell basically it divides through mitosis. It undergoes mitotic division to make two haploid nuclei called the male nuclei. Okay. And this vegetative cell also has one nuclei. Now this vegetative cell plays a role in forming this pollen tube. Okay. And inside the pollen tube, there is a vegetative nucleus and there are two male nuclei. Is it clear, Arpit? Can you see the diagram?
Yes, sir.
And why are there two male nuclei? Because I just told you that for the formation of zygote, the fertilization event that we call in angiosperms, it is known as the double fertilization event or the double fertilization. Why it is called double fertilization? Because remember there were two male nucleus.
Yes, sir.
I have used here the male nucleus 1 and male nucleus 2. So the first male nucleus goes and fuses with the egg cell. Okay, both are haploid, so it makes a diploid zygote, which develops into a diploid embryo, and ultimately that embryo becomes like seed. It's present in the seed, and then that seed grows. Now this another male nucleus, which is a haploid, goes and fuses with polar nuclei. Now, I told you that we will discuss polar nuclei and endosperm, but before that, we have to understand one more thing, which we will do today. It is called the embryo sac. So write down. Let's start with today's class. Did Mehraj join back? No. He lost. Okay. Embryo sac. Now, what does the word sac means?
Pouch-like structure.
Yes, a bag which carries something. Now, clearly the name suggests what it is going to carry.
Embryo.
Embryo.
Embryo. Now, the embryo sac in angiosperms is known as a 7-celled and 8-nucleated structure. Okay, everyone? Sometimes they ask you in question that what is the 7-celled and 8-nucleated structure in angiosperm? So that is the embryo sac. Now let's draw this and let's try to understand the embryo sac. So clearly it says it has 7 cells. So the whole structure, the embryo sac, contains 7 cells total. But there are 8 nucleus, which means that any one cell will have more than one nucleus, right? You have to fit 8 nucleus in 7 cells. So some cell has to take up an extra nucleus. Clear or not? This is just basic logic.
Yes, sir.
So,
Yes. So let's try to make our embryo sac. It's oval in structure.
sir, this embryo sac will be in the ovary, right?
Yes. Yes. In the ovary, I told you there are ovules which become seed, correct?
Yes, sir.
There, it's present in there. So, and I also told you that in a ovary there is a micropylar end. Remember? Sorry, in a flower there is a micropylar end. Let me make a small. So this is a stigma. Let's say this is the style, and then there is a ovary. Correct?
Yes, sir.
And I told you when a pollen lands, it makes a pollen tube, and this pollen tube goes all the way down, and inside there is this ovary present, right? Suppose this is the ovary. Now this pollen tube does not enter the ovary right from the top. It goes all the way around and enters the ovary from here. Remember I told you this? And this side is known as micropylar end. Okay. Why it is called, the word micro is associated with the male.
Small.
Microspore and macrospore. In last,
Micro is small.
micro means small, but here the small, the word micro. Okay, I'll come to that. Probably you did you remember? I told you the microspore word is associated with pollen, and macrospore word is associated with egg cell in plants. Okay, if you do not remember, write it down right now so that you never forget. So this macro word will always be associated with the female, and the micro word will always be associated with the male. Okay, microgamete and macrogamete. In humans also, sperm is very, very small as compared to ovum. Do you understand? So sperm is the microgamete and ovum is the macrogamete. Here also, the egg cell is the macrospore or the macrogamete, and the pollen is the microspore or the microgamete. Is it clear? Fatima, Arpit, and Alia?
Yes, sir.
Yes, sir.
Okay, very good. Now it goes all the way around and enters from here, which is known as the micropylar end. This, so in an embryo sac also, let's come back here. In embryo sac, there are 2 ends. This is the micropylar end. So it's clear that from this side, the male nuclei will enter, right? And the other end is called the chalazal end. Okay.
So what did we do till now, sir?
Yes, Mehraj. I was just thinking you will join back. Nothing, you have not lost anything. We have just started with embryo sac. I just told that you were there when I was telling about double
Okay.
fertilization event. Now just pay attention to
Okay.
embryo sac is a structure in angiosperms where it contains the embryo, it develops into the embryo. It's called embryo sac. It's a 7-celled and 8 nucleated structure. Okay, and we are drawing it right now. So it has a micropylar end and a chelazal end. Towards the chelazal end, there are 3 cells. Okay, there are 1, 2, and 3 cells present towards the chelazal end, each containing a nucleus. And these 3 cells are called antipodal cells. This diagram may also come in your boards where you have to make it and explain it. So there are 3 cells, antipodal cells. Towards the micropylar end, there are again 3 cells. Okay, but let's not draw it like this. So 2 cells are towards the front, which are known as synergid cells or synergids. And just behind the synergid cell, there is a 3rd cell with the nucleus. And this is a special one. This is the egg cell. So if the question comes, the egg cell in an embryo sac is present towards which end? So what will you write?
Micropylar end.
Yes, micropylar end. Now these 2 synergy cells also has 1 nucleus. So now tell me, till now, how many cells are there and how many nucleus are there?
6.
6 cells and 6 nucleus are done, right?
Yes, sir.
We are only allowed to go with 1 more cell, which is the central cell, and this is the biggest cell. It is known as the central cell, and it has 2 nuclei. Okay, and they are called polar nuclei. They are both haploid. Polar nuclei. Okay, both are haploid, but 2 haploid nucleus are there. And this cell is
Excuse
called.
me, sir.
Yes, please.
Sir, isn't this polar nuclei a small structure?
Polar nuclei is a small structure.
Oh, sir, my teacher made it and it was
Nuclei is small, but the cell in which the nuclei is present is the biggest cell. So central cell is the largest cell of the embryo sac. Okay. These nuclei are small. This is not to scale. I'm just giving a diagrammatic representation. Do you understand?
Yes, sir. Yes, sir.
Yes. So I can make it small, but the problem then will be, or should make it small. There are 2 nuclei. Can you see it now?
Yes.
Did it work? Happy, Arpit?
Yes, sir.
Okay, good. If you are happy, I'm happy. So this central cell contains 2 nuclei, which are called the polar nuclei. And towards the micropylar end, where the synergids are present, there is another thing called the filiform apparatus. There is a structure known as filiform apparatus. Now this is the complete overall diagrammatic representation of an embryo sac in angiosperms. Okay, embryo sac, angiosperms. Now, if a pollen tube is going to enter, let's say this is the pollen tube which is coming, so it will enter from this side, right? Pollen tube will enter from this side, and the pollen tube is containing the male nucleus. Remember I told you this?
Yes, sir.
So this is the male nuclei. Now one of this male nuclei will go and fuse with the egg cell, this cell. Do you understand now the double fertilization here? Male nucleus 1 fuses with the egg cell, which is present towards the micropylar end, just behind the synergids. Is it clear? And the other second nucleus goes and fuses with this polar nuclei. So now tell me, what is the ploidy of polar nuclei?
When?
No, it's still N.
En.
No, No, it's n plus n. It's not 2n. It's 2 haploid nucleus. So it will be n plus n. Do you understand, everyone? If 2 haploid nucleus are present together, but still they are distinct, different nucleus, we will still call them haploid. They are not diploid. Do you understand? A diploid is where in one nucleus there are 2 copies present. Say yes or no. Do you understand or not?
No, sir. Can you repeat?
Yes, I will. Now this polar nuclei is not 2n. It is n plus n structure. Because there are 2 haploid nucleus present inside this big central cell. Polar cell we also. Do you understand this?
Yes, sir. Yes.
Now in one nucleus, let's say this is a nucleus. In this nucleus there is only one copy of the genome. That will be called n. We call a nucleus 2n when there are 2 copies of the genome. One from the mother and the other from the father. So this structure will be called 2n. Okay. If there are 2 nucleus where there is single copy and single copy, it will be n and n. It still will be haploid nucleus. Is it clear? This is what I was telling you, Arpit.
Yes, sir. Yes.
So polar nuclei is not a diploid nuclei. These are 2 haploid nucleus present together. Do you understand, Fatima, Alia, Mehraj? If there is any doubt, please ask me.
After the fertilization of the polar
We are coming to that. We are coming to that. Now, so this is this clear? N plus these 2 n's coming from the polar nuclei, both the polar nuclei. So how many total n's are there?
3.
Now, if these 2 cells, look here. Now, if this male nucleus and these 2 polar nuclei, if they fuse in the process of fertilization, they will give rise to endosperm, which will be what will be the ploidy of endosperm? Now they are all fusing.
3n.
Yes, 3n. So do you understand, Fatima? Now, I think this was your question.
Yes, sir. Yeah.
You did not understand?
No, I understood. Yes, sir.
Okay. So n came from male nucleus and 2 n's came from each polar nuclei. So there are 2 polar nuclei. So how many nuclei are fusing together? How many nuclei are fusing together in this second
3.
fertilization event? 3. So because 3 nuclei are fusing together, this second event is known as triple fusion. So the question which has come in board CBSE is, what is double fertilization and triple fusion? So this is triple fusion. Okay. Is it clear, everyone? Triple fusion is a part of double fertilization. Is it clear to everyone? Say yes or no. Then I'll move.
Yes, sir.
So please make this diagram and complete this thing right now. I'll just make it a little small. Can you see this? Sorry. Can you see this now?
Yes.
On one screen.
Sir, we earlier did this in our 10th class. I remember.
This one, the double fertilization.
Yes, sir. So the second event is, I guess, known as single.
Yes.
And embryo sac also we did.
Yes. Now we have to read about these cells also. But before that, done? Let me know when you're done.
Sir, done.
Okay. Fatima, Alia, Maraj.
Yes, sir. I'm done.
Now let's come to, yes, Alia's done. Let's come back and finish this thing that we left. Clistogamy and chasmogamy. Write down. So you understood that for a pollen to reach this egg cell, it has to make a pollen tube, and this is how it reaches the egg cell, right? Now, apart from, on the basis of whether it is a self pollination or a cross pollination, like in with respect to the genes, we call we can divide pollination into autogamy, geitonogamy, and xenogamy. Now, clistogamy and chasmogamy, they are on the basis of the structure of the flower. Okay, so write down clistogamy first. Clistogamy. The word cleast is associated with closed. Something like a fist. Okay. So the structure of a closed fist, said cleast. And gamy, you know, union. Okay. Now write down. In clistogamy, the flowers which are produced by the plant, the flower which are produced by the plant do not open up. Do not open up. Okay, whereas you can just write down the second thing that Arpit brought into today's class, chasmogamy. It is opposite to clistogamy. It means in this, the flowers which are produced by the plant open up. Flowers are opened. Is it clear, Arpit?
Yes, sir.
In a way, chasmogamy can be, in a chasmogamous plant, there can be autogamy, geitonogamy, or xenogamy all happening. So this is on the basis of the process from which anther to which stigma the pollen is getting transferred. If it is getting transferred from the same flower to the same flower, it is autogamy. From between two different flowers on the same plant, it is geitonogamy. And if between two different plants, it is xenogamy. But all these three examples, autogamy, geitonogamy, and xenogamy, for this, at least for geitonogamy and xenogamy, the flower has to be open, correct? Yes or no?
Yes.
Yes, sir.
If the flower is not open, geitonogamy and xenogamy cannot happen, but still autogamy can happen, correct or not?
Yes, sir.
So now do you understand that clistogamous plants are strictly autogamous? Yes or no?
Yes, sir.
This is just a different criteria to distinguish between types of pollinations. Remember in biology, I told you, no, I did not tell you, that whenever we are doing any kind of a distinguishing or we are making types, it is on some basis. Like I can say types of organs in my body. Now that can be on the basis of which organs are present in the head region, in the abdomen region, in the thorax region, or on the basis of which are organs associated with circulatory system and non-circulatory system. Yes or no?
Yes, sir.
Whenever I say type of something or classes of something, it always have to be on some basis. Like on the basis of the structure of nucleus, there are two types of organisms, prokaryotic and eukaryotic. Remember? Yes or no?
Yes, sir. Yes, sir.
Yes. So that type is on the basis of nucleus, but on the basis of whether the cell has cell wall or not, there are other types of organisms. Yes or not? Right? Plants have
Yes,
cell
sir.
wall. Animals do not have cell wall. Bacteria and fungi have cell wall. So types always come with some basis. So do you understand that chasmogamous? Now let's come here. So tell me, a chasmogamous plant, can a chasmogamous plant be autogamous?
Yes, sir.
Can
Yes.
it be gitonogamous?
Yes,
No,
sir.
sir.
No, Fatima? Why? Why do you think?
Because these do not open, right?
Chasmogamous. Chasmogamy flowers are open.
Then yeah, they can.
So if a flower is open, anything can happen. Self also, cross also between the different flowers on the same plant also. Auto, gitono, xeno, anything can happen in chasmogamy, right? So write
Yes.
down, in chasmogamous flowers, both self-pollination and cross-pollination is possible. Both self and cross-pollination possible. Whereas in a cleistogamous flower, can gitonogamy and xenogamy happen in a cleistogamous flower?
No, sir.
No. So only thing which can happen is,
Autogamy.
yes. So write down, only self-pollination is possible because they are closed. Now do you understand that the example which you gave me here, P is for autogamy, P undergoes autogamy, but P as a plant produces closed flowers. It produces cleistogamous flowers. So P is an example of cleistogamy also. Is it clear?
Yes,
It's
sir.
a cleistogamous autogamous plant. Now for chasmogamy, any open flower can be an example, right? Hibiscus. Do you understand, Arpit?
Yes, sir.
Get out clear?
Yes, sir.
Okay, let's move forward then. Now, so what is clear? Embryo sac, yes. Now write down, in formation of zygote, write down some points. So I'm back to this second stage, okay? Formation of zygote. The first stage was pollination, second stage is formation of zygote. In between, we also studied cleisto and chasmogamy. It is not a part of the stages, okay? It belongs to pollination, the first stage. I hope it's not confusing, people. Everything is sorted?
Yes, sir.
Good. Now write down about the second stage. The zygote which is formed upon fertilization, the zygote which is formed upon fertilization is located near the micropylar end. Makes sense, right? Because egg cell is near the micropylar end, so the zygote will also be near the micropylar end. Correct? This was a neat
Sir,
question. Yes, Arpit?
sir, what is the meaning of chelazal?
What is the meaning of
Chelazal end that you wrote up.
can you chelazal end?
Yes, sir. Yes, sir.
It's just a term given to this end. Like chalaza is a structure about which it will become very complicated then. So because I'm not discussing chalaza here today, but just know that micropylar end and chalazal end are two ends for now.
Okay, sir.
The chalazal tissue, if we go into detail of this, the class will get diverted and we will not be able to finish what we intend to do. You can read upon chalazal tissue if you find any difficulty. So you will not be asked on this. I've never seen a question on this, but if you really want to know, you read, and if you found any difficulty, come back to me. Then we'll discuss. Or I can discuss at the end of the class. Let me first finish this. 2 minutes. You just stay. I'll discuss this. Okay.
Okay, sir.
Yeah. So the egg cell is towards the micropylar end. So the developing zygote or the developing embryo or the male nuclei, it will all enter from micropylar end towards the micropylar end. Now write down the zygote starts dividing. Why will a zygote start dividing? To make a embryo, right? Write down the zygote starts dividing only after some amount of endosperm is formed. Okay, so this thing, the transition from a zygote to embryo, this thing here will only start when endosperm is produced, when some amount of endosperm is present. Now this already gives you a hint that what is endosperm there for, what is the function of endosperm. Any idea? No idea? Okay, write down endosperm is the tissue which is responsible for providing nutrition to the developing embryo. Endosperm is a tissue which is responsible for providing nutrition to the developing embryo. So now do you appreciate why double fertilization happened? Yes or no? Arpit, you are mute. Fatima, why did double fertilization happen? To ensure that one fertilization will make zygote, but when that zygote will develop into embryo, for division, for cell division and for growth and development, energy is required, nutrition is required. Yes or no?
Yes, sir.
Where will that nutrition come from? So
Endosperm.
endosperm, not endoderm. You said endosperm or endoderm?
Yeah, I said endosperm.
Yes. So this endosperm is a triploid tissue, and this is it's a best fit to provide nutrition, because more copies of genome means more protein formation. Yes or no? I told you how does protein forms in every cell. The central dogma, remember? DNA makes RNA, RNA makes protein. Remember? People, no? So if there is more DNA content, there will be more protein formation also happening. So this triploid tissue will be more nutritious. It will produce everything in greater quantity and can also provide nutrition to embryo, apart from providing nutrition to itself to survive. Okay. Now let's study about endosperm in detail. Write down endosperm. I hope everyone is understanding. Please keep saying something or discussing. When you go blank, I do not understand whether you are understanding or not. Endosperm. Write down. It is the nutritive tissue. It is the nutritive tissue which is present in the seed along with the embryo. Nutritive tissue present in the seed yes, Arpit. It is the nutritive tissue present in the seed along with the embryo. where is it present? For that we have to go to study the seed. So we now come to the third stage. So I'm holding up endosperm here by just telling you what it is. And to study this in detail, let's first go to the third stage. So the third stage of sexual reproduction in flowering plants, the last stage now, is formation of seed and fruits. Pollination resulting in the formation of zygote, which makes embryo, then formation of seed and fruit. Is it clear? Finally everything ends at seed and fruit. Okay, so yes or no?
Yes, sir.
So write down few things about this stage. As you know, ovule becomes seed, and ovules is present in the ovary, which contains the embryo sac. So ovules become seed and ovary becomes fruit. Now you all have seen seeds and fruits both, right? But there are certain fruits which are seedless, correct? All the fruits do not have seed. Yes or no?
Yes, sir.
So fruits that do not have seeds, then how do they propagate? If they do not have seed, how do they propagate? The answer is simple. They propagate through vegetative propagation. Remember, plants can undergo vegetative propagation. Arpit, Fatima, Alia,
Yes, sir.
wake up. Now let's first talk about seed, then we'll go to fruit. Now for a seed, there are two types of plants. One is called monocot plants. Have you heard this word before in junior classes? And the other is dicot plants. Now these two plants you have already studied in plant kingdom chapter, remember? So to study this, revise this. Is my voice lagging for you people? Because I think it's lagging for Arpit, he's writing it to me. Fatima, is my voice lagging for you as well?
No, sir.
No?
No.
Probably it's your network then, because it's not lagging for Fatima. Okay, so to understand monocots and dicots in detail, please go to your 11th plant kingdom chapter in NCERT and revise it. But one difference is in the seed structure in these monocots and dicots. Now this word monocot and dicot comes from one structure which is called cotyledons. So they are also known as monocotyledonous plants. Now the word mono means,
One.
yes, single, and the word cotyledonous comes from cotyledon. Now what is a cotyledon? To understand that, in a dicot they are called dicotyledonous, which means they have two cotyledons. Now to make you understand cotyledon, let me make a seed here. So how many of you have seen a gram seed, also known as chana?
Yes, sir.
Kabuli chana. So it looks, let's say it looks something like this, right? It looks something like this. It's not exactly. And it can be split into two, correct? You can
Yes.
split the chana into two halves here. So you can open that seed, correct? It's like a joint seed, you can open it from the middle. Now, these two things that you are seeing, which are opening up, are cotyledons. When the chana is raw, like I'm talking about, have you seen the green raw chana, green raw gram seed. You can just split it, and if you look carefully, there is something present at the top here, right? A knot like thing. When you put these gram seeds in water and soak it in water, they start sprouting. So you see structures growing from the top in these chana seeds. Am I correct?
Yes, sir.
This sprouting happens from this chana seed. This is the developing embryo. The sprout is actually the developing embryo which is coming out of the seed now. Is it clear? Yes or no?
Yes, sir.
So if you go and put that sprouted seed in the soil, it will germinate into a new chana plant, a plantling, and then a whole plant, correct? Now, when you can open this into two, there are these two cotyledons. In the cotyledon, what is filled is the endosperm. So the cotyledon contains the endosperm tissue. Is it clear? And we are eating the endosperm when we are eating a raw chana. Do you understand now? So if a seed has two cotyledons, it is called dicot. If a seed has only one cotyledon, for example, you also must have seen so gram seed is a dicot. Have you seen a rice seed or a corn seed? Like rice and corn themselves. If you see a rice or a corn, rice is just like, it's like this kind of a grain, right? You cannot split rice into two. Can you split rice into two?
If you try to, you can, but naturally you cannot.
No, you can break it into two from here, or you can, it's very difficult to break it into two from here, but it cannot be opened up like as easy as chana can be just split into two.
Yes, sir.
You can see that it is already two cotyledons stuck together. It's not one structure, but the rice seed is one single structure. You can break it into pieces, but you cannot split. You cannot split its cotyledons. You understand what I'm trying to say, Fatima?
Yes, sir.
Yes. Same thing is for a corn. So you see a corn looks like this, right? Something like this, and it's just one thing. It's slightly whitish towards this region and yellowish towards this region, right?
Yes, sir.
So a corn or a rice are examples of monocots. Is it clear?
Yes, sir.
Now let's draw the structure. So for a monocot, if I draw a monocot, let's draw corn. So in a monocot seed, because there's only the seed itself is the cotyledon, it has only one cotyledon. The major half part of the seed actually is divided, and it contains endosperm. Is it clear? The embryo is present in this other half. Now this embryo has different tissues which are lined like this. First layer yes, Arpit. Can you hear me, Arpit? If you can hear me, you can stay with the class. If you are finding it difficult to hear and understand me, then it's up to you, okay? But please make sure that you get this, the recorded lecture. Otherwise, you will lag behind. Arpit is finding some problems with the internet. Anyway, okay, so do you understand? This part here is the embryo. Okay, and just above the embryo, the tissue is called scutellum. This is from monocot. And one characteristic feature of a monocot seed is that the endosperm is lined with layer. And this layer is called alluron layer. So monocots are alluronic seeds. Monocot seeds are alluronic seeds. One question that was asked. Will you remember this, Fatima?
Yes, sir.
Okay. So draw the structure first. Same we will do. Can I delete this, Fatima? This part.
Yes, sir.
So on this side, we can actually draw the dicot. Okay. Now, a dicot seed. Let's make. So this is or a dicot. Let's make a bean. Rajma, things like that. They are also dicot, right? Kidney bean, let's say. This is how a bean seed. So I'm splitting the bean seed. Do you understand? These two are cotyledons. Okay, Fatima?
Yes, sir.
And inside the cotyledon, if you talk about where is endosperm, so endosperm is present in both the cotyledons. So where is the embryo then? The embryo is present here. It's like a structure which grows later on. This is the embryo. It's kind of sandwiched between the two cotyledons. That's why when we put a chana to sprout, between the two cotyledons, this sprout comes out. This sprout is actually the embryo. Is it clear?
Yes, sir.
Yes.
Now, why am I only the one in the class?
Good question. I think so. Arpit is having issues with the internet. He's not able to stably stay connected. I don't know about Ali and Miraj. They also.
Also, sir, the embryo will be only that much, or it will be until the end?
Yes, it is going to be very long. Like it completes the whole part. So if you say if you mark the embryo, this whole cotyledon, the size of the embryo is this whole cotyledon size.
Okay, thank you.
Yeah. In monocot, it is only in the lower region. The upper has endosperm.
Yeah, sir.
Now write down parts of the seed. Parts of a seed. This is important. Whether it's a monocot or a dicot, some parts are common parts of the seed. So there are three parts which you see. Okay, I did not make. So for both the seeds, the outermost thing is the seed coat, that you know, right? For any seed, whether it's monocot or a dicot, the outermost layer has to be the seed coat, correct?
Yes, sir.
And then there has to be endosperm, and the third thing which is important is the embryo. There are three things: seed coat, endosperm, and the embryo, correct? The most important three parts. So write down parts of seed. There are three parts of a seed: seed coat, endosperm, and embryo. This is where we will discuss all in detail, more depth. So the first part, seed coat. If you have seen a gram seed, so a roasted chana, it has a covering on the top, right? Which you can just take out. It is rigid, hard covering, which we generally do not eat.
Yes, sir. Yeah.
Yes, that is the seed coat.
So it's there even for avocado, right?
Avocado seed, the seed of avocado you're talking about.
Yeah.
Yes, it is. Seed coat is present in every seed. Even for a mango seed, if you just put a mango seed and dry it, you will see that first when you break it open, it opens, and from inside the cotyledons will come out. They are greenish inside.
Oh, yeah, I saw something like that. Yeah.
Yes. So if you break a mango seed open, inside there also something present. That is the seed. The outside one, which is very fibrous. When you eat a mango, there are many fibers attached with the seed, right?
Yes, sir.
Fibrous. So that fibrous structure, that is the seed coat. Okay, so you understood what is seed coat?
Yes, sir.
Now seeds come in all different colors, sizes, and they are round, wrinkled, hairy. Hairy, by hairy I mean that mango seed, fibrous. They have fibers attached. Or for the most part of their life, a seed is dormant till it finds a good environment to grow. Okay. Have you seen seeds? So let's take an example of the chana seed. The chana seed will not sprout unless we soak it in water or with like wet cotton or a wet cloth, correct?
Yeah.
So what happens when we put it in wet cotton? How does the seed comes to know that now is the right time to grow up, versus when it was kept in a bottle, the seeds were not growing up?
Because the water has some minerals.
But the seed inside, the cells inside, if the seed inside is alive, it already had endosperm. Like if you're talking about nutrition, some minerals, the endosperm is full of nutrient, water, and mineral, right? This is
Yeah.
not dried up. A seed looks dried from outside, but still inside it has living cells, right?
Yes, sir.
If a seed completely is dehydrated and desiccates, it can never grow, right? Can it?
No.
So living cells has to be inside, and if a cell is
Can
living,
you make a seed completely dehydrated?
yes, if you just keep a seed forever, like it gets dehydrated and it then dies, and then this only this endosperm is present and we eat it. Like a roasted chana, when you roast it or when you bake it over the heat, or a pop, like a corn, when you put it on fire, it just pops. Why? You know why does it pop? Because it has moisture inside. Where is that moisture inside? A corn seed has a lot of moisture inside, right? Because it is living, it will be having cells and tissues, and cells will be having cytoplasm. And what is cytoplasm? Fluid.
Yes, sir.
Yes. Yeah. So how do you think seed emerges? The embryo comes out while sprouting, and it was not coming out for a very long time when the conditions were not adequate. Now for this part, I will take you back to class 11th. You remember you read something called imbibition in the diffusion chapter? Imbibition, remember? Imbibition is when solids like wood absorb water and then they swell up. They have lots, they swell up because water is filled inside. Remember imbibition?
Yes, sir.
If you put a wood in a crack of a rock, which is very fitting in that crack, and you leave it over the monsoon, when it will imbibe water, it will have so much of hydrostatic pressure that it will, create larger cracks because of that pressure. This was an example given in NCERT only, class 11.
Yes, sir.
Now the seed coat also behaves like that. When this seed coat imbibes moisture when it is put in moisture. This is why whenever we germinate a seed, we put the seed and then we pour some water. Now that water is not the immediate water that the embryo needs. It is the water that will first makes the seed coat, imbibe it, and then because of the hydrostatic pressure, the seed coat will break apart. And when the seed coat will break apart, only then the embryo can come out. Is it clear, Fatima?
But after the seed coat is broken, why does the embryo still need the water? Because the endosperm will be providing the nutrients, right?
Yes. So for some initial time, the seed actually does not use the water for itself. Actually, the endosperm is providing ready-made nutrition. Because how does a plant makes food? Through photosynthesis, correct?
Yes, sir.
But when the first embryo, the sprouts come, are those sprouts green in color?
No,
What color are they?
they're yellowish.
Whitish. Sprouts are white.
Oh, sprouts, yeah.
Yeah, the sprouting part is white, which clearly tells that it does not have chlorophyll yet, right?
Yes, sir.
Which also tells us that it cannot photosynthesize yet. So
Yeah.
it has to depend on the endosperm. Okay.
Yeah.
But water, as the cells will keep dividing and new things will be forming, the nutrition is coming from endosperm and water is coming from the ground, but photosynthesis is not happening yet. So the first contact with moisture of the seed is for the seed coat to imbibe the water and break apart. This breaking of the seed coat tells the embryo that now the environment is favorable because there is water around, which means now I can come out, and the seed coat is also broken. So now I have a way to come out. Before that, even if the embryo wants to come out, it cannot come out because it's closed inside the seed coat. So write down first about seed coat. Seed coat protects the internal parts of the seed. Seed coat protects the internal part of the seed. And this seed coat itself has two layers. Right now, seed coat has two layers. The outer thick layer is known as testa. And the inner thin layer is called tegumen. Okay, so the seed coat itself is a double layered structure. Outer thick layer is testa and inner thin layer is tegumen. Is it clear, Fatima?
Yes, sir.
Yes. Now, another thing, it prevents, seed coat also prevents loss of water, right? Because inside there is endosperm, there is embryo, and all these are living structures. So they will be having water in their cells, right? But if a seed is just lying there, the heat, through heat and evaporation, it can lose all the water and then can get dehydrated and die. So the seed coat also protects the loss of water or dehydration.
So, but eventually, if the water, if the water is not provided, still it will get dehydrated, right?
Gets dried up, yes. But if there was no seed coat, it will get dehydrated even fast. So there are many seeds from which you can remove the seed coat. So you can do this experiment yourself. You bring some rajma seeds, okay, or any bean. You bring. Have you ever helped your mother with taking pea seeds out from the pea pod?
Yeah.
When you take, yes, very good. Now it's easier to make you understand. So pea is a dicot, okay? So first there is this pod, right?
Yes.
You open the pod, inside the pod there are seeds, right?
Yes, sir.
Now you take one seed out and you will see that seed will be attached. So there will be one attachment mark. It will be attached to the pod through some structure, right?
Yeah.
From there you can actually rip the whole seed coat.
Ah.
Yes. If you rip the whole seed coat, inside you will find two cotyledons just stick to each other.
Yeah.
And then they can very easily get separated if there is no seed coat.
Yes, sir.
It is round in structure because of the seed coat. It looks one very smooth, continuous structure. But if you remove, just peel the seed coat out, there will be two cotyledons, two hemispherical cotyledons stick to each other like this. On the top of it was the seed coat. So you keep a chana, a seed with the seed coat somewhere, and you keep one without the seed coat where there are these two hemispheres just attached to each other. And you see which one dries off faster. Which one will dry and desiccate faster?
Without the seed coat.
Yes. So now does it make sense? How does it help?
Yes, sir.
Yes, right. A lot of science can be done with. I believe that a lot of science is done in kitchen actually.
Yeah.
Our mothers just don't know that they are scientists in action. They are actually practically doing what we are studying. You know, when we go and study about baking soda, about how salts ionize, how does it forms, how does it loses electron, how does it makes bond, and things like that. They're actually doing that same thing in the kitchen every day, correct?
Yes, sir.
Yeah. So what have you written? What was the last line?
It also prevents loss of water.
Yes, it prevents loss of water and it also prevents parasites from entering into the seed, because if a parasite enters, it might damage the embryo and the seed will die. But they cannot protect it always. Sometimes you will see that you open a pea seed and from inside it is damaged.
Yes, sir.
If a parasite enters somehow inside the seed through the seed coat, it will damage the embryo inside. But seed coat provides protection against parasites.
So how do the insect go inside this through the seed coat?
Yes, very good question. I was discussing this in last class, with, in one of the batches. There was a program that used to come, and I used to watch it on Discovery called Monster Inside Me. Have you heard of this program?
No.
Monster Inside Me. So that program used to talk about organisms or parasites or these pathogens, insects which have infected a human and are growing inside the human skin or somewhere inside the organ. So what happens is there is a wasp, and I'm not sure if I'll be able to make a wasp beautifully. So wasp has very big eyes, and then it has something to suck the nectar, right?
Yeah.
And they have very small thorax and they have a big abdomen, right?
Yes, sir.
They have 3 pair of legs, and some of these wasps have a very long ovipositor, a very long thing present at the tail. It is coiled in structure. Now this thing, it doesn't look like a wasp at all, but pardon my drawing. But this is called ovipositor. Ovi means egg, positor means to deposit. So these structures are used to deposit eggs. So they can sting basically. So they can, just like a needle, penetrate when that seed was developing. You know, sometimes, has it happened to you that sometimes a fruit looks very okay, fine, fresh from outside? You cut the fruit, inside there is a worm.
Yes.
Have you ever wondered how did that worm enter there?
So I see some holes in the fruit.
Some very minute holes, right? But the worm is always bigger than those holes.
Yes.
So when that worm was egg, like it was not larva, it was egg, it was at the egg stage, these insects deposited those eggs in a fresh and soft fruit. And over time, as the eggs hatched, they were around the endosperm. Everywhere there was nutrition. So they were just feeding in that fruit, on that fruit. Is it?
Yeah. Yeah.
So this
I
is
get
how these insects and parasites enter the seed. The parents deposit them inside these seeds. Interesting, no?
Yeah.
These wasps can also be very dangerous to human, because sometimes they can, if a human is sleeping, so there have been incidences where humans are sleeping outside the house or in a picnic somewhere around the jungle or something, or even in their houses, and these wasps just like it will feel to you like they have bit you, and it will start itching. So just near the soft tissues, like beneath the ear, if you touch your ear, like touch the part of the ear which is beneath this ear pinna, you see there is a soft hole type of structure where the lower jaw is attached to the upper jaw. Do you see this
Yes.
here? So sometimes they bit, the wasps bit here or here around the temple, and you feel like it's just a wasp or a bee bite sting and it's okay. But they actually deposited the eggs. And over time, these eggs will start developing as larvae inside the human tissue. And that program used to tell about these things that, and hence the name Monsters Inside Me. Okay,
Yeah.
it's possible. But these are different types, different species of wasp. They are not the yellow wasps or bees which are normally around us, because when they sting in self-defense, and their sting, like a bee sting, stays attached to our skin sometimes.
Meaning?
So this is the abdomen of a bee. Okay, a bee abdomen. This is the body and this is the head. Okay, this is the bee abdomen.
Yeah.
So a bee stings from the abdomen, it does not stings from the mouth, you know that?
Yes.
Yes. So in that abdomen there is a needle like sting like this, correct?
Yes, sir.
So when it penetrates this sting inside our skin in self-defense, this stays inside the skin, and when it takes out the abdomen and flies away, it gets detached.
So doesn't that like make some problem for the bee
Yes,
also?
it does. It does. It does. So a bee can sting technically only once. The same bee cannot sting twice if it has lost the sting. Okay. And it does, it's like a wound, no? It's like an open wound with that bee. But if the bee manages to get away and survive, then it will heal itself. But the sting stays. So most of the time, if someone gets bitten gets stung by a bee, look carefully if there is a sting still left there. Do not rub that structure or do not put something over it, a cloth or something tightly. That, because that sting might need to be removed. Okay.
Yes, sir.
So much of science happening around us, no, Fatima? My
Yeah,
God.
it's so interesting.
It is. And biology is the kind of science on which we are also living, right? We are living biological laboratories.
Yeah.
Everything is happening inside us, so it becomes even more interesting. So seed coat, yes, write down. The seed coat, also the next point, the the seed coat also prevents the embryo from germinating. The seed coat also prevents the embryo from germinating during unfavorable conditions, during unfavorable environmental conditions, during unfavorable conditions. Okay. So I think I gave you 4 or 5 points about seed, its importance, no, of seed coat and its importance.
I wrote them in a paragraph.
Okay, cool. No problem. Try to write things as bullet points. You know, it will help you as a student when you will go back and revise. It will make it easier for you to revise. I always used to make notes as bullets. I never used to write paragraphs. Only in examinations I used to write paragraphs, not in my notebook. It helps a lot. Okay. What was I saying? Yes. So a seed coat also prevents the embryo from coming out in like in a unfavorable condition, because see, unless and until the environment is favorable, so if there is moisture around, the environment is favorable, correct?
Yes, sir.
If there is no moisture around, the environment is not favorable for an embryo. Even if the embryo manages to come out in a dehydrated, very hot environment, will it survive?
No.
It will die. So the seed will waste itself, right?
Yeah.
This is where seed coat comes into play. So the seed coat will not break until there is water and moisture around, because it breaks by the principle of imbibition. It imbibes water, it gets swollen up, and because of the pressure it breaks. Okay. Once it breaks, now it's a proof that the environment is favorable. There is water and moisture around, so the embryo can now germinate. Is it clear?
Yes, sir.
So till it is not favorable, it prevents the embryo from coming out. About the seed coat, there is one question that I have seen. There is a structure known as hilum. 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 Write down about this. It was just a question. So write down. It is like a small opening. Write down. Hilum is like a small opening in a seed towards the micropylar end. Remember micropylar end? When it was an embryo sac, then happened fertilization, then became zygote, embryo, and the seed.
Yes, sir.
So hilum is like an opening towards the micropylar end, and this structure, it is visible in many seeds, especially in dicot seeds. And this structure was asked in NEET because it is equivalent to, right now, it is equivalent. When we say equivalent, it means a comparative structure. It is equivalent to navel in humans. So navel is the belly button. In our bellies, there is an opening to which umbilical cord was attached when we were inside the mother's womb. That is called navel, right?
Yes, sir.
So this part actually is an opening in our body. After we are born, the doctor put 2 clips, right? And then cuts the umbilical cord from the center close to our belly. Over time, that small part of the umbilical cord dies and sheds off, leaving our navel hole slightly, like prominently, it's like a dip in our belly. Just beneath that, there is a cavity. You know that? What is that cavity called? Just beneath the navel. So if technically, like when if I take a scalpel or a blade and starts like clearing the tissue from the navel, like if I keep poking inside, I will reach in the body cavity. Is it clear, Fatima?
Which body cavity?
We will reach the opening of our belly. We will reach inside the belly. Have you seen Matrix? The movie Matrix? They put a.
No.
Have you seen or not? Yes or no?
No.
No. Oh, okay, news. Simply it means that we can open the, like, it is a very weak point, and from the navel you can actually reach inside the abdomen. Okay, it's like an opening,
Okay.
just like this small opening which is at the micropylar end of the seed. So hilum is equivalent to navel in humans. Write down where the umbilical cord is attached. Where the umbilical cord is attached. There is a cavity just below the navel in humans which is called hernia. As a doctor, you will be hearing about hernia a lot. Hernia is a problem. We think that hernia is a disease. Lots of pain happens in the stomach, in that lower abdomen, below the navel, because there is a cavity. This is the cavity, hernia. Okay?
Yes, sir.
Just telling you these things you should know. Now remember hilum. And the other parts are endosperm and embryo, which are the major parts. Okay, so about which we will study in the next class. Okay, Fatima, we'll complete parts of seed, then we'll complete fruit, and we'll try to wind up plant reproduction. Then we'll come to human reproduction. Okay, Fatima.
Okay, sir.
Okay. See you in the next class. Bye
Thank
bye.
you. Have a nice day.